KURINJI Senior Secondary School, CBSE, Namakkal.
Successful result producing school in state every year.
Administered by the Board of Directors with over 35 years of experience in the teaching field.
Special attention on art, craft, music, dance, silambam, yoga, skating and karate.
Focus on Hindi Sabha and Spell Bee Olympiad exams.
Smart class for effective and innovative learning and well-furnished library.
Solitary importance on sports and games in state and national level.
WhatsApp facilities to know activities of students and school inside the campus.
Free bus for LKG to 8th standard.
Free, hygienic and nutritious food.
NEET, JEE, IIT coaching experts from Rajasthan and Andhra Pradesh along with our effective teachers.
Admission registration for classes LKG to +2.
KURINJI Educational Institution, Kavetipatti, Namakkal. Admission in progress. CBSE, KG to 12, contact 9025895176. Metric 6 to 12, contact 9344567484. For online admission, log on to www.kurinjischoolnkl.in.
Okay students, so now in this video, we are going to discuss regarding vectors for cloning genes in plants as well as in animals. So here in this video, so before start to learn this video, already in previous video I have given information regarding cloning vectors, isn't it? In cloning vectors, what is information we learned? So we have learned regarding the features of plasmid we learned now. So there what are the points we learned? So definitely suppose if you would like to take any kind of a plasmid as a vector, so that particular plasmid must consist of must have following features. Likewise we learned there origin of replication, selectable marker, and one more is cloning sites. Likewise we have taken. Now 3 completed, 3 side headings, and 4th one now we are going to study. So continuation of cloning after cloning vectors. Next side heading is vectors for cloning genes in plants as well as animals. So this concept important, try to concentrate on this. Now that information is transferring genes into the plants and animals. One more time, transferring of which one genes from the plants as well as animals, from the bacteria and viruses. So now in this particular case, what is the point now I am going to take? So what is the point now? Transferring genes into the plants as well as animals from bacteria and viruses. Means if you would like to transfer, so if you would like to transfer any kind of a foreign gene, or if you would like to transfer the foreign genes into the plants as well as animals, definitely we need to take the help of bacteria as well as viruses. So which have known this for genes. So how to deliver genes to transfer the eukaryotic cells and force them to do what the bacteria and viruses want. So for example, so here what is the information I have written now? Definitely in order to transferring of a foreign gene, so into the plants as well as animals, so definitely we need to take the help of bacteria as well as viruses. So in this particular case now, so here one important point or one important example now I am going to take. What is it? Agrobacterium tumefaciens. Agrobacterium tumefaciens, it is a pathogen of several dicot plants. So don't forget, so Agrobacterium tumefaciens is a pathogen of several dicot plants, is able to deliver, it means it is able to deliver a piece of DNA known as tDNA to form normal plant cells into tumor. So here what is information? Agrobacterium tumefaciens, it is a pathogen. Agrobacterium tumefaciens. Once you see if you observe that Agrobacterium tumefaciens scientific name, no, so there we will find it is a bacterium. So means it is a bacterium and it is a pathogen, means it will cause diseases in organisms, means where it will cause diseases in dicot plants. So means this one means Agrobacterium tumefaciens now we are using for what? To deliver a piece of DNA. So means suppose if you have any kind of desirable gene, so that particular desirable gene we can deliver by using of agrobacterium tumefaciens. So what is the DNA fragment we will deliver through the agrobacterium tumefaciens? That particular DNA or special characters DNA or foreign DNA, so we will be calling as tDNA to transfer normal plant cells into tumor cells. So suppose when this particular tDNA is entered into the plant cells, when this particular tDNA enters into the normal plant cells, what will be happened means normal cells are converted into tumors. So in this particular case, what is the point? So agrobacterium tumefaciens, one bacteria is there. So that particular bacteria, even though that particular bacteria is a pathogen, so definitely we will be using that particular agrobacterium tumefaciens for delivery of our foreign gene. So means what is a foreign gene? We will be incorporating into the organisms. That particular our foreign gene or desirable genes are the characters which are good. So that particular DNA we will be calling as tDNA. So this particular tDNA now we will be transferring into the normal plant cells. So why we are doing or transferring like that means, so we would like to get the tumor cells. Why? Sir, tumor cells means what is the benefit? Why we are doing, sir? Why means by introducing of this particular tRNA, why we are changing this normal plant cell into tumor cells means, what is the meaning? No. When one particular normal cell is converting into tumor cells, no, so definitely what is the information we have to take is definitely normal plant cells definitely converted into tumor cells. So thereby, so before when these normal cells are going to be converted into tumor cells, no, our foreign gene means what is a tDNA we are incorporating into the normal cells, no, so that particular thing will be multiplied. So that is the point here we have to understand. That's why when our foreign gene is going to be multiplied, normal cells are going to be converted into tumor cells. Or how we can remember means, so when these normal cells are going to be multiplied, so means the group of cells or the bulk of cells formation takes place and appears like tumors. So in this particular case, these particular tumor cells, each and every particular tumor cells, definitely consist of our foreign gene. So that one, and directly these tumor cells to produce the which one chemicals required by the pathogens. So definitely these particular tumor cells, by using of these tumor cells we can produce chemicals which are required by the pathogens. So likewise, so somewhat information means here in this particular case agrobacterium tumefaciens we are taking. So in that particular case, this particular pathogen can cause diseases in dicot plants. So even though we are using this agrobacterium tumefaciens for delivery of our foreign gene, that particular gene we will be calling as tDNA. So thereby what is happening means normal plant cells are converted into tumor cells. So when normal cells are going to be converted into tumor cells, in tumor cells, so we can find out more number of tDNA. So why? Because when plant cell is going to be divided, tumor cells of formation takes place more and more. So indirectly, what is the point we can understand means our foreign gene means what is the gene we have inserted newly now. So that particular gene is multiplied in the tumor cells. So similarly, so means here agrobacterium tumefaciens here we are using. So agrobacterium tumefaciens here we are using, so for incorporation of our foreign gene so into the plant cells. Suppose if you would like to transfer our foreign gene into the animals means what we have to use, sir, means here you see retrovirus. So retrovirus definitely we can be used so in animals. So in order to transfer of our foreign gene, so into the animals means retrovirus definitely we can be used. So that is only information I have written. So similarly, retrovirus in animals have the ability to transfer the normal cells into cancerous cells. Don't forget in plants, in plants what are the cells of formation taking place? tumor cells of formation taking place, whereas in animals, so by using of retrovirus, cancerous cells of formation now we are seeing. Don't forget, in plants agrobacterium tumefaciens, so here one point I didn't write on the board, but that point I will give you. What is that point? No, this particular agrobacterium tumefaciens consists of TI plasmid. So TI plasmid, I hope I will get you will get this information here. Okay, right. So this particular agrobacterium tumefaciens consists of TI plasmid. Exactly, let us spotlight you see. So TI plasmid. Okay, that information when I will come here I will give you. Now let us discuss there. Similarly, retrovirus. So if you, in order to incorporation or in order to transfer of our foreign gene in animals, definitely what is a virus means what is organism we can use means retrovirus we can use. So for want to transfer the foreign gene. Okay, so now the ability, the transformation of the normal cells into cancerous cells. So when due to the activity of retrovirus, so when we will incorporate our foreign gene into the normal cells, no, these normal cells are now finally converted into cancerous cells. Don't forget, cancerous cells information we are studying in animals and tumor cells information we are studying in plants. So don't get confused, important. Next one, these tumor cells, tumor inducing TI plasmid of agrobacterium tumefaciens has now been modified into cloning vector. Now, so tumor inducing means TI plasmid of agrobacterium tumefaciens. So within the agrobacterium tumefaciens, now what is a plasmid is present? Now TI plasmid is present. TI means tumor inducing. So this tumor inducing plasmid, so definitely is modified into cloning vector. Sorry, suppose this agrobacterium tumefaciens is a pathogen, no. So then why we are using, sir, this one means, so why we are using this plasmid means, so here definitely we can use this plasmid. Why? Because now we are changing. So what we are changing, this particular TI plasmid character, so somewhat we are modifying. So this particular agrobacterium tumefaciens now have been modified into cloning vectors. So koncham means somewhat slightly we are changing that particular agrobacterium tumefaciens as cloning vector, which is no more pathogen. So here once you see that agrobacterium tumefaciens we have taken as a pathogen first, but when we will be giving of some kind of modifications, the same vector only or the same pathogen only, now it is not acting as now it is not acting as a pathogen. Did you remember this one? Try to listen carefully. So initially same agrobacterium tumefaciens we have taken as pathogen, but when we will be giving of some kind of modifications, okay, so some kind of a modification, so automatically what is happening, that now that particular pathogen now it is not acting as pathogen. So but it can be able to use mechanisms to use in the mechanisms and to deliver the genes in our interest into the a variety of plants. Means here genetic characters means what are the characters or what are the disease causing characters are there, no? By modifying that particular agrobacterium we are eliminating the which one pathogenic nature. So once pathogenic nature is everything is left out, so that particular agrobacterium tumefaciens we can be used for the transfer of our foreign gene. So likewise here somewhat information they have given. Next one, similarly retrovirus information also have been disarmed, means here, and are now used to deliver the desirable genes into the animal cells. So definitely before we learn, so retrovirus definitely used for the incorporation of our, the transfer of our foreign genes. So in this particular case, now what is information? So this particular retrovirus definitely disarmed pathogen, means here, so here in a retrovirus what are the characters or what are the genes are responsible for the for acting as a pathogen, no? So such kind of characters we will remove. So means when that particular retrovirus have been also have been disarmed, no, in that particular case, no, we can use this retrovirus for delivery of our genes, for delivery of our genes. So here in this particular case, once this particular retrovirus is disarmed, so that particular virus we can be used for the delivery of our desirable genes into the animals. Don't forget, important. So once the gene or the DNA fragment has been ligated to the suitable vector, means here this particular retrovirus or TA plasmid definitely we are using for our transformation of for the transfer of our foreign gene. So that particular gene or the fragment, now we will be attaching with one suitable vector. It is transferred into the bacteria or the plant cells or any kind of animal host where it multiplies. Means, so by using of retrovirus or by using of TA plasmid which is present in the agrobacterium tumefaciens, definitely, so what we can be done means, definitely that particular foreign gene now we will be attaching with the suitable vector, that is maybe that suitable vector may be retrovirus or that suitable vector may be TA plasmid, we will be giving attachment. So later that particular suitable vector now transferred to the bacterial cell or the plant cell or animal cell, whatever may be the host, we will be taking for incorporation of our which one recombinant DNA into the host. So host is nothing but where it multiplies, means our recombinant DNA is there. Again, recombinant means, you don't forget, now till now our foreign gene is there, that one we are incorporating with the one particular plasmid. So when this particular plasmid receives our foreign gene, so definitely that particular plasmid we will be calling as recombinant DNA. That particular recombinant DNA, already I told you, recombinant DNA is nothing but, so or it is consist of which one, two different types of genes together. So that is the meaning of recombinant gene. Now competent host, this explanation is important, listen carefully. So in a competent host means here we need to create one competent host. So here why? Because so actually here what is information, I will give you some more information orally, later information concerned I will explain. So competent host means actually we are going to insert which one, recombinant DNA or the recombinant DNA. So in this particular case, when we are going to when we would like to incorporate one foreign gene, so into the host, host will not show interest to receive the which one, recombinant DNA. Why? Because here that particular recombinant thing is nothing but DNA. Now you can follow. So that is since DNA is a hydrophilic molecule, now here the DNA here is a hydrophilic molecule. So thereby it cannot pass through the cell membrane. See there. So that's why, so when this particular DNA is acting as a hydrophilic molecule, it cannot pass through the cell membrane. That's why, sir, this particular DNA, when it is not going to be or not entering through the cell membrane, then how we can transfer this particular recombinant DNA into the cell, sir, means, so in the next information they have given. So in order to force the bacteria, so now one more time, in order to force the bacteria to take up the plasmid, now we are putting somewhat pressure on the bacteria. So actually, so bacterial cell wall is there now, that particular bacterial cell wall is like a cell wall is digested by the lysozyme enzyme. So I think did I give this point or not, I don't know, but so definitely bacterial cell wall can be digested by lysozyme enzyme. I think so I have given in some other time. Now you see there. So in order to force the bacteria to take up the plasmid, so now we are forcing the bacteria for what? To receive the plasmid. Now the bacterial cells must be made competent to take up the DNA. So now here it must have the ability. So which one? Host. Host must have the ability to receive the which one? DNA. So means it must become as competent. So then only it can be take or it can take. So this is done. So how that particular host can be or can become as competent means, so now key information is there. This is done means what is can be done? So means normal host that can be converted into competent one. So how, sir, that one become a means? Now you see there. So this is done by treating them with the specific concentration concentration of divalent cation treatment such as calcium ions. Now here when we are going to do that, or when we are going to treat, or when we are going to give the treatment with the specific concentration of divalent cation with the calcium members, which increases the efficiency with which DNA enters. So thereby by using of competent, means by using of a specific concentration of divalent cation, means when we will be using of divalent cation treatment, divalent cation, my example calcium, now we are taking here. So automatically what will be happen now when we are going to do that divalent cation treatment with calcium members, definitely we can increase the efficiency. So with which the DNA enters, means here definitely we can increase the efficiency to receive the foreign DNA into the bacterial cells. Now a bacterium through the pores in its cell wall, the recombinant, so here you see that increases the efficiency with which the DNA enters the bacteria to the pores in its wall. So thereby the recombinant DNA can be forced into such cells by incubating the cells with the recombinant DNA on the ice and followed by placing them briefly at 42 degrees centigrade. That is only we will be calling as a heat shock, and then putting them back onto the ice. This enables the bacteria to take up the recombinant DNA. So here this information I read, I think so you cannot understand, but now you don't see the board. Now just you concentrate on my voice. So now I will explain. Now once you see, now first of all, what is the thing we are doing? Divalent cation treatment we are doing with calcium. So now when we will be doing or when we will done this divalent cation treatment, which increases the efficiency with which the DNA enters into the bacterium through the bacterial cell wall or bacterial pores. When we will be doing of divalent cation treatment, automatically what will be happen? That particular bacteria cell wall will be converted into pores, means here, so some kind of pores formation takes place on the bacterial cell wall due to the divalent cation treatment with calcium. So when some kind of pores formation takes place on the bacterial cell wall, automatically at that particular time, so we need to do that, or we need to continue that process. So what is that? In this particular case, that particular cell wall or that particular bacteria, now we are keeping on the ice. Once you see that. So here you will find, so force them incubating onto the recombinant DNA on the ice. Now it means here one more important point, when pores we are finding on the bacterial cell wall, that particular bacteria, and beside to that particular bacteria, now we have to place the recombinant DNA also. Both now we will be placing on the ice. So later once we will keep these two things, means one is recombinant DNA and one more is bacterial cell. Okay, so when we will be incubating, when we will be keeping or incubating, so and on ice, so automatically what will be happen? So definitely these two members, means recombinant member or a recombinant DNA trying to enter into the bacterial cells only after the completion of these two steps. Means so first of all divalent cation treatment we will do. Later when we will be keeping a recombinant DNA and bacterial cells on the ice, later followed by heat shock treatment at 42 degrees centigrade, and again we will be keeping on the ice. Means first of all bacterial cells, these bacterial cells we will be placing on ice, along with the bacterial cells we are placing recombinant DNA on the ice later. So second step, so what is the thing we are doing now? Heat shock treatment we are doing at 42 degrees centigrade. Again later we are placing, so this particular entire procedure we are shifting onto the ice. So thereby what will be happen? So first of all ice treatment, later heat shock treatment, again later ice treatment. So this enables the bacteria, so last line you see, so this enables the bacteria to take up the recombinant DNA. When we will be placing or when we will be doing, means first of all ice treatment, heat shock treatment, later ice treatment, automatically, so that particular recombinant DNA definitely enters into the bacteria. So means here, sir, for each and every time this is only a process, sir, for both plants and animals means. So here one more methods, a few methods are there. So what are those? No, this is not only the way to introduce the alien DNA or foreign DNA into the host cells. Not only this way means which one? Ice treatment, heat shock treatment, and ice treatment. Not only this one, not only this one. One more thing also is there. That is so few methods. So now first method is microinjection. So in this microinjection, what are the things we will be doing means the recombinant DNA is directly injected into the nucleus of an animal cell. So here this one means here the microinjection step so is meant for the animal cells. So in order to, or if you would like to do that any kind of, or if you would like to do, or if you would like to incorporate any kind of a foreign gene into the animals, so definitely what is the best method you can follow? No, that is microinjection. So by using of a microinjection method, the recombinant DNA is directly injected into the nucleus of animal cell. So this point is important for NEET examinations. Next one more alternate method, another method is there suitable for the plants. So means one procedure means microinjection procedure suitable to the animal cells. Now one more alternative method is there, that is for plant cells. Now these plant cells are bombarded with high velocity. So when we will be bombarding these plant cells with high velocity, automatically, so these microparticles, velocity microparticles of gold and tungsten coated with DNA, so in a method known as biolistic method or gene gun method. So here in this second method, so definitely this particular second method we can be used, suppose in order to transfer of our foreign gene so into the plant. So what is the method we can be followed? That is biolistic method or gene gun method. So means here in that particular method, on that gene gun method, so microparticles are present. So these microparticles are made up of gold as well as tungsten. So on the microparticles, now we will be keeping, you see there, on the tungsten as well as gold particle coated with DNA. So means on that particular microparticles, we will be giving a coating with DNA. So when we are going to operate the trigger of that particular gun, so that particular microparticles will be released from that gun and enters into the plant cells automatically. So that particular microparticles consist of DNA particles, DNA particles. Okay, so that particular DNA particles now enters into the plant cell. So likewise, by using of biolistic method or gene gun method, we can incorporate our DNA into the plant cells. Means in plant cells, a good method is a biolistic method or gene gun method. Next one, in animals, microinjection is best method. Next one, last method also is there. That one is disarmed pathogen. So vector, which when allowed to infect the cells and transfer the recombinant DNA into the host. So means definitely disarmed pathogen also means here the one particular pathogen is there in that particular pathogen. So by removing of disease causing characters, so definitely so we can use that particular pathogen for the transfer of our foreign gene into the host cells. So thereby, so likewise there are different types of procedures we can be seen. So in order to incorporation of our foreign gene into the host cells. So here some more information is there. So if you are not understanding this concept, definitely I will explain by one more time in presence of you. Now let us move on to the, so till now we have learned the information regarding tools. What are the tools which are required, or what are the tools which are required to the process of recombinant DNA we have seen. Now let us have a look on the processes of recombinant DNA technology. Now what is information now? So the process of recombinant DNA technology involves several steps in specific sequences. So several steps are then in specific sequence means this particular specific sequence we should not skip. Okay, specific sequence such as, so try to by heart these steps one by one. Specific sequence try to by heart. So first one is ligation of DNA. So first of all isolation of DNA. So, first of all, that is isolation of DNA. Second one is fragmentation of DNA by restriction endonuclease. Next one, isolation of a desirable DNA fragment. Next one, ligation of DNA fragment into the vector. Next one, transferring the recombinant DNA into the host. Next one, culturing the host cells in a medium at a large scale, and extraction of desirable or desired products. Means likewise there are different types of things are there. Now without reading, now let me explain. First of all, so what are the procedures of recombinant DNA technology? Now first of all, we need to search for the desirable gene. So once you will find the desirable gene, so from the entire gene, we will isolate the DNA. So later, so by using that particular DNA, now by using of restriction endonucleases, we will do the cuttings. Means so first of all, isolation of DNA. Later, so now when you will find the DNA, so that particular DNA we will isolate. Later, by using of restriction enzymes, we will cut into mini pieces. Later, so isolation of a desirable DNA fragment, means isolation of a desired DNA fragment we will do. Later, ligation of a DNA fragment into the vector we will do. Later, transferring the recombinant DNA into the host cell we will do. Later, culturing the host cells in a medium at a large scale, and extraction of the desired product. Okay, man. Now here in a competent host, so what is information we will be learning now? So in a competent host, generally, so in order to incorporation of our foreign gene into the host cells, definitely, so first of all, ice treatment, heat shock treatment, as well as ice treatment we will do. So before this process, definitely divalent cation process we will do. So later in animals, microinjection process. Next one in plants, biolistic method or genic method, and later and last method, disarmed pathogen. So likewise there are different types of processes are there in order to incorporation of our desirable genes into the host. Next one, processes of recombinant DNA consent. So first of all, we have to search for the DNA. Once the DNA you will find that particular DNA you have to isolate. Later we have to cut that particular DNA into the fragments. So why? Because for obtaining of our foreign gene, so with the restriction enzymes. So later once you will find the DNA fragments, all these fragments, now you have to separate based on the molecular weight. Later one desirable DNA we have to get by the process of gel electrophoresis. Later that particular desirable DNA we have to ligate or we have to associate with the vector. So that particular vector, now it will become as a recombinant. That particular recombinant DNA now we have to incorporate into the host. Once our foreign gene or the recombinant gene is entered into the host, now that particular host that must be cultured in the medium. So once that particular host cell is cultured in that particular medium, automatically in a large scale, so we can extract the desirable products or desirable antibiotics or desirable good products from that particular desirable DNA. So likewise there are different types of processes are there in recombinant DNA technology. So in the next video, definitely let us learn the information regarding processes of recombinant DNA technology. Thank you.